Directional variability in visual examination, introduction and core concept. The research paper Directional Dynamics Reveal Regimes of Human Visual Examination introduces a novel metric for understanding how humans inspect visual information. The authors, Jose Navas and colleagues, propose that traditional eye tracking measurements which primarily quantify how far and how fast the eyes move miss a crucial dimension: how viewing direction changes over short time intervals. This limitation is significant because eye movements during tasks like medical image interpretation involve complex patterns that cannot be fully captured by simple magnitude-based metrics. While existing approaches analyze fixation sequences and spatial patterns, they typically operate at the level of aggregated fixations rather than capturing the moment-to-moment directional adjustments that occur during ongoing examination. What is directional variability? Directional variability is defined as the variability in angular change between consecutive gaze displacement vectors within a short temporal window 10 to 50 milliseconds. To compute this metric, the researchers track the direction of eye movements between consecutive samples, calculate the angle between successive movement directions, and then measure the standard deviation of these angles within a window. Behavioral interpretation: high directional variability, frequent directional corrections within constrained spatial regions characteristic of sustained local inspection where small corrective displacements maintain foveal positioning. Low directional variability, directional persistence during transitions between spatially separated locations consistent with SACCADIC movements between regions. This metric is independent of movement magnitude, meaning it isolates changes in orientation rather than distance traveled, providing a geometric description of how viewing direction evolves over time. Key findings: 1. Stable bimodal distribution. Across two large datasets, Reflex 3052 radiograph examinations and gaze-based 12,334 general viewing recordings, directional variability consistently exhibits a robust two-mode bimodal distribution. This structure persists across temporal windows from 10 to 50 milliseconds. In contrast, traditional metrics like velocity, acceleration, and step length variability show asymmetric spike plus tail distributions without comparable mode separation. The separation strength for directional variability was approximately 4 to 10 times larger than for magnitude-based metrics. Cohen's d equals 3.40 versus 0.49 to 0.85. Importantly, the bimodality reflects within subject dynamics rather than pooling across heterogeneous observers. The subject variance ratio analysis showed that only 1 to 4% of total variability is attributable to stable between subject differences, confirming that individuals dynamically alternate between regimes during viewing. 2. Regimes reflect directional organization, not just spatial extent. The researchers tested whether directional variability regimes simply reflect spatial dispersion. They found that directional entropy, angular reversal density, and return rate differences between regimes persist and in some cases strengthen after controlling for spatial extent through quartile matching. For example, in Reflex, directional entropy effects increased from d equals 0.78 to d equals 1.19 when spatial dispersion was controlled, a 52% increase. This demonstrates that directional variability captures directional organization independent of spatial spread, distinguishing it from metrics that primarily partition by spatial extent. 3. Predictive value beyond traditional metrics. At the study level, directional regime composition predicts scan path topology metrics and reporting time beyond comprehensive magnitude-based baselines including velocity, acceleration, and mean turn angle. For 20 millisecond windows, transition entropy prediction improved from r squared equals 0.024 to r squared equals 0.116 delta r squared equals 0.093. Unique spatial cells visited, from r squared equals 0.059 to r squared equals 0.128 delta r squared equals 0.069. Self-loop fraction, from r squared equals 0.236 to r squared equals 0.300 delta r squared equals 0.064. These predictive gains remain stable across temporal scales 10 to 30 milliseconds windows and were statistically significant after false discovery rate correction. Theoretical and practical implications. Oculomotor interpretation. High directional variability windows are compatible with sustained periods of local inspection, during which small corrective displacements maintain perceptual stability. Low directional variability windows reflect directional persistence during SACCADIC transitions between spatially separated regions. Rather than isolating individual microsaccades, these 10 to 50 milliseconds windows capture the net directionality of multiple small displacements. Clinical applications in radiology. The Hitlov regime high directional variability, low velocity most closely corresponds to sustained local inspection characterized by frequent directional corrections within constrained spatial regions. This provides a quantitative framework for describing examination strategies previously characterized qualitatively, e.g. Driller vs. Scanner behaviors in volumetric imaging. Potential applications include radiology education, characterizing examination behavior patterns. Expertise research, differentiating novice and expert search strategies. Human-AI interaction, incorporating directional statistics into attention models. Relationship to existing theories. Directional regimes extend established frameworks including focal versus ambient processing Hoffman, 2016. Global versus local search in radiology. Exploration versus exploitation in visual foraging. However, unlike heuristic threshold-based approaches, directional variability provides a data-driven, continuous statistic for identifying examination modes without predefined task phases or region of interest boundaries. Limitations and future directions. Several important limitations are acknowledged. Expert-only samples, the radiology dataset comprised only expert observers, preventing conclusions about novice-expert differences or training effects. No diagnostic accuracy validation, regime composition was not validated against detection performance or diagnostic accuracy. Dataset level boundaries, regime boundaries were estimated at the dataset level, not validated for individual level inference. Spatial discretization, topology metrics relied on a 10x10 grid, alternative continuous representations may provide complementary insights. Future priorities include, validating relationship between directional regimes and diagnostic accuracy. Assessing individual level reliability through test-retest analysis. Extending to mixed expertise cohorts and additional imaging modalities. Exploring continuous or graph-based topology representations. Conclusion. Directional variability represents a distinct, behaviorally informative dimension of gaze dynamics that captures how frequently viewing direction changes within short temporal windows. By identifying regime structure directly from directional statistics rather than heuristic thresholds or predefined task phases, this approach provides a data-driven geometric framework for characterizing examination dynamics. While clinical utility requires further validation linking regime composition to performance outcomes, directional metrics offer promising applications in expertise research, visual search modeling, and human-AI interaction.